So now we are going to discuss about Kala's culture. Right. Are you able to see this slide?
Yes, sir.
Right. Is it clear?
Yes, sir.
Right. So today topic is Kala's culture. Already told you yesterday what is Kala's how the Kala's formation. The Kala's formation by any explant by culturing process. So during this process, undifferentiated group of mass of cells are formed. This group of cells, undifferentiated group of cells called as a callus, right? Here the explant may be root, stem, leaves, whatever it may be, any part of the plant can be utilized as a explant, right? So after formation callus, we should culturing the callus. Let's see the what is the process. Yesterday we have seen the cell culturing, right? So cell culturing, what type of medium we can utilize here for cell culturing? We can utilize only the liquid medium, suspension medium. It is called a suspension medium. But the callus culture we can use solid medium, mostly solid medium. But sometime we can use the liquid medium also. That is based on the formation of callus. If the callus compactly formed, we can use the solid media. If the callus form just loose formation tissue, in that situation we can use the liquid medium, a liquid nutrient medium. So anyway, most probably for callus culturing, we can use solid medium. And I already told you how the media preparation. Again, we discuss about the media preparation also. So here, uh, where it is, just wait. Right. If you have book, turn on page number 35. Turn on page number 35, callus culture. Right. Just wait. So let's see steps in callus culture. The same steps involved in the tissue culture techniques, the same steps involved in this technique also. For successful initiation of callus culture, there are some important steps are utilized, followed. The first one, aseptic preparation of plant material. Before that, actually, we selected explant. First, explant selection. Explant. What is explant? What is explant? Explant means any part of the plant called as explant. So first actually before aseptic condition, first of all we selected the explant material, right? After explant material, we provide the aseptic condition. So how we provide aseptic condition? During aseptic condition, we sterilize the surface of explant with help of HgCl2, mercury chloride, and sodium hypochloride, and also some detergents, and by finally washed with distilled water. So that is the first step. So in this process, we create a aseptic condition or aseptic preparation for the plant material. So before that, you select the explant. After selected the explant, you sterilize them with help of the HgCl2, sodium hypochlorite, so on, right? And finally washed with distilled water. After that, selection of nutrient medium. Selection of nutrient medium with appropriate ratio of auxins and cytokinin. What are the ingredients involved in preparation of medium. Already told you the first class. So for preparation any medium, here the required some macronutrients, macro and micro, macro and micronutrients also involved required, macro and micro. And apart from here, the in callus for callus culturing, the major role by phytohormones. Major role by phytohormones. Here the phytohormones may be the phytohormones mostly auxins and cytokinins. But when compared with when compared with cytokinin, the auxin ratio should be in high. So that is the important feature of the callus culture. So callus culture, we utilize the auxins, such as auxins may be high concentrated auxins such as IBA, indole butyric acid, or naphthalic acetic acid, NAA, IBA, NAA. Actually, these NAA, IBA, this type of auxins mainly for root formation, mainly for root formation, called as a rhizogenesis root. For this auxins, for this high concentrated auxins, when you add cytokinin, when you add cytokinin, cytokinin, now shoot formation. Now shoot formation form. Shoot formation. Are you able to see? Hello? Are you able to see the marker?
Yes, sir.
Right. So these are the required for the preparation of the medium. After that, actually, before incubation, what we have to do before incubation, that means you prepare the explant with sterilized explant and also prepare the medium also. That what would be the next step? Now explant ready to produce the callus and also give supplement nutrition. Here the nutrient medium also prepared. After that, actually the third step should be, the third one should be transfer of explant, transfer of explant. That means transfer of aseptic explant into the medium. That is the third one. So the first step, explant prepared with sterilized explant. This explant transferred into the media, transferred into the sterilized media, How the nutrient medium sterilized? Already we have learnt during practical training classes.
Yes, sir. In incubation.
Not incubation.
Autoclave.
Right. In autoclave. After preparation media, we keep in autoclave for sterilization. And here the explant sterilized and also autoclave and also media also sterilized. Now the explant introduced into the media. After that, this is called inoculation. After inoculation, now finally the fourth one, final step, incubation of culture media, controlled physical condition and proper optimum temperature. So where it is incubated, which instrument utilized for incubation of the media?
Laminar.
Right, laminar airflow. After finally we shifted to the laminar airflow for weeks. And next step. So these are the steps in callus culture. Is it clear? What happened after after 4 to 6 weeks? Callus formation from the explants, callus formed. So I already told you the callus is the group of the cells. The callus may be compactly formed or loosely formed. If it is compactly formed, utilize the agar media. If it is loose formation, utilized liquid media, right? Is it clear?
Yes, sir.
Next, let's see the factors in culture media. Factors. So let's see the factors affecting callus culture. Many factors affect the callus formation in vitro culturing. Maybe condition of explant first. Second, physical factor. Third, growth regulator. Already we discussed it just now. Again, so first, condition of explant, physical factors, and growth regulators. Right? So first condition, explant. The callus formation actually is not information. The callus formation is influenced by the explant nature and the genotype and age of the genotype or age of the explant. Maybe some explant may be young, some are old. So we can select the young explant or old one also. Under genotypic condition also, we take the consideration genotype. Genotypic condition means gene makeup of the plant, particular plant. This is also very important for the preparation of colors. And next one, physical factors. Physical factors may be temperature and also light, and also media. Media also one of the physical factor. Media, nutrient media also physical factor. Let's discuss one by one. First, temperature. Temperature range for the callus culture should be 22 to 28 degrees centigrade. This is optimum temperature for callus. So we regulated the temperature at 22 to 28 degree centigrade. Second one, light. Actually, the light may be utilized for some species, but some species dark without light. So that is the based on the species. It is based on the plant species. Light may be essential or dark may be essential. So that is depend on species. So anyway, using light and dark, 12 hours. 12 hours. If you the uh, light species, light dependent species, the light utilized for 12 hours. Or it is dark also dark related species, the dark utilized for 12 hours. So this is physical factors. And last one, media. Media also another physical factor. So actually, I already told you generally strong auxins utilized. Already told you strong auxins. The highly strong auxins are 2,4-D and 2,4,5-T. These are the highly strong auxins. 2,4-di and 2,4-tri. And apart from the auxins, there's a little amount of auxins, cytokinins also utilized for the media preparation, right? So these are the affecting factors. And next one, growth regulators. The growth regulators in the media strongly influence the callus formation. Cellular differentiation and organogenesis is mainly depend on the concentration of auxin and cytokinins. From the beginning, have been telling that about this. So for organogenesis or morphogenesis should require the two major auxins and cytokinin. Auxins and but in especially in auxins, highly concentrated, highly strong auxins utilized such as 2,4-D and 2,4-T. And this is the factors affecting the callus culture, right? Actually not given in our book. That is the applications importance of callus culture. So from the callus mainly the somatic embryo formed. From the somatic embryo, we can develop the whole plant. So from the callus can generate the whole plant. That is the one use of the callus culture. Next, a very important one is study of biosynthetic pathway of various metabolic process by using trace element in callus culture. From studying the studying cells, we can study the biosynthetic pathway. Biosynthetic pathway means C3 cycle, C4 cycle, Krebs. These are all any type of cycles, any type of pathways, any type, and also lipid metabolism and fat metabolism in plants, GOGAT, amino acid formation. These are all we can study in callus observation, in callus study. So, but for this we can use some of the tracer elements in the callus culture. Next, another important one is production of secondary metabolites. Production of from the callus culture, we can produce secondary metabolites such as alkaloids and flavonoids, so on. So this is about callus culture. Is it clear about callus culture? First definition callus. First definition callus allow.
Yes, sir, it's clear.
Right. So definition callus and steps in callus culture, and next affecting factors of callus culture and utilization application of the callus culture. And next one we see, right.
Callus culture is important.
Yes, any type of culture important. First of all, you should read the first unit, tissue culture techniques. Tissue
Yes, sir.
culture technique. Tissue, because the the rest of the units, further units, all depend on the tissue culture unit. If you understand about the tissue culture technique steps, the rest of the all units will be easy to you. That's why this is very important, tissue culture techniques and also application of tissue culture. Tissue culture techniques steps and application. Second unit also very important. Second unit, they are all very important, right? If you can develop the concept of the tissue culture techniques, the rest of the all clonal propagation and cell culture and callus culture, organ culture, there will be easy to you. Well, the small changes, for example, cell culture, difference between cell culture and callus culture. The simple difference is in cell culture, we mostly, not mostly, only we use the liquid media, suspension media for callus culture, for cell culture. But callus culture, mostly we use solid medium, agar medium, MS media, solid mainly MS medium. That is the different. But rest of the process same. Rest of the process means in the steps also, in the steps, for example, cell culture also we select the explant. After that, production of callus. But here one different is after production of callus, this callus introduced into the conical flask like this conical flask and transferred to the orbital shakers. Transfer the orbital shakers. Due to the shaking, the callus cell separated. But in callus culture, we don't use any orbital shakers. We cannot treat any agitation to the culture media. But in cell culture, we should agitate the culture media. Agitate means that means the it some create some pressure to separate the cells. So that is the small changes, but rest of the all same, right? That is the small difference between cell culture and organ culture. And first of all, if we see the diagrammatically, For surface sterilization, that is the first step already told you. Select the explant. After selection explant, surface sterilization with help of the chemicals HgCl2, sodium hypochlorite, so on. Finally, washed with distilled water. And the second step, what is the second step? In second step, preparation media, agar media, preparation of agar media. So agar media also prepared. After preparation of agar media, this is the this explant transferred into the media, Next, incubated. Later, and also the callus, after formation callus, after incubation callus formed. After formation callus, subculturing of the callus into new media. Subculture of callus, new media. The new media may be that means already told you if this callus after formation callus, the group of callus, if it is compactly like this compactly formed, if the callus formed compactly, we can use solid media. But instead of that, this callus formed as a loose, like this. In this situation, we could use liquid media, right? So this is about callus culture. Next one, protoplast culture, I think, right?
Yes, sir. Protoplast culture.
Right. Protoplast culture important. Protoplast culture important. Let's see protoplast culture. Right. So what is protoplast first? Already told
Plant
you
cells
in
without the cell wall.
right. Plant without cell wall, the rest of the portion called as protoplast, right? So here protoplast, just wait. Cells without the cell wall, right? Without the cell called protoplast. So without cell wall, what are the next layer? If we separate the cell wall, what is the next layer? Next layer should be plasma membrane. Plasma membrane and other cellular components. What about other cellular components? What does it mean? Hello?
Yes, sir.
First of all, tell me the cell components already told you during cell biology genetics in
Yes,
core
sir.
subject,
Mitochondria,
right?
then Golgi apparatus, chloroplast.
Right. But you have to tell in order. First of all, cell wall, first layer, plasma membrane, second layer. After plasma membrane, with plasma membrane called as protoplast. Without plasma membrane called protoplasm. In protoplasm, nucleus and all cell organelle present. If you separate the nucleus, that is called as a cytoplasm, right?
Yes, sir.
For example, this is the cell wall, plasma membrane, nucleus. The same cell wall, plasma membrane, nucleus and surrounded by cell organelles. But here without nucleus, only organelles, rest of the organelle present, that is called cytoplasm. That is the difference between protoplasm and cytoplasm, right? Is it clear? Hello?
Yes, sir.
Right. So here in this process, in protoplast culture, we can culturing the both protoplast of the different species. So this is one protoplast, and we take another different species protoplast. We can mix with both to the media transfer the media, incubation, finally get plants. We will discuss later about this. And that process called as somatic hybridization. That is called somatic hybridization. But be careful when they ask question, direct question where they can ask about protoplast culture or somatic hybridization somatic hybridization. Another one somatic hybridization. And next one hybridization or hybrid formation, hybrid formation. So in this only normal protoplast with enucleated, that means nucleus absented, enucleated protoplast, which means this is nucleated protoplast, but here no nucleus. If this both are fused, one fruit, this is called hybrid. This is called hybrid. Is it clear? Somatic hybridization, hybrid. Hello?
Yes, sir.
Right. So
So, sir,
somatic
for
hybrid.
both protoplast culture and somatic hybridization are same.
Same.
Okay.
What is involved in somatic? Soma means vegetative. Here only vegetative cells involved in the form of protoplast. This is protoplast and this is also protoplast from not same species. Of course, we can use the same species also, but for better healing, better characters, better desired character, we can select different species, right? So that is somatic hybridization. Hybridization means one is protoplast, another one is cytoplasm. Protoplasm fused with cytoplasm, that is called hybrid. Is it clear? Hello?
Yes, sir.
Right. So, okay, protoplast culture, okay, hybrid, okay, but how we collect the protoplast? We know where is protoplast, just to remove the cell wall, the rest of the content called as protoplast, right? Hello?
Yes, sir.
That means for collection of protoplast, just we remove the cell wall, right?
Yes, sir.
Right. So let's see the isolation of protoplast. There are two methods to isolation of protoplast. One is mechanical method. And second one is enzymatic method. First, let's discuss about mechanical method. So this mechanical method utilized for only some cells. Some cells means there are some proper characters should have. The cell should be in large and cell should be in highly vacuole with the storage tissues. Only this type of cells we can utilize it for mechanical isolation. The large cells, high vacuolated storage tissue placed in osmotic solution. That means hyper solution. Placed in hyper solution. This is beaker, high concentration solution. Uh, this is the cell placed in cell. What happen if the a normal storage cell placed in high concentration? Here the liquid concentration high, but cell concentration low. What happened? The water flow inside the cell or flow outside the cell. Hello?
Yes, sir.
If the normal cell placed in hypertonic solution mean high concentration. Here high concentration. Tell me the simple one. First of all, this is layer W water, S sugar. Now tell me the water can move for sugar or sugar can move for water.
Water can move for sugar.
Right, water moved. So here the water molecule high, here the less water molecule. That's why the water moved toward the S. S for high concentration. Right? Now tell me, here high concentration. That means water high concentration. Here only low concentration inside the cell. Now tell me, the cell water
Water move towards the cell, sir.
water
Move
inside
towards
the,
the cell.
that means cell absorb the water or cell release the water to out.
Observe the water.
Where is high concentration? That means first of all, water means this is low concentration. Here sugar means high concentration. Always water move from the low concentration to high concentration, right?
Yes, sir.
But in this diagram, where is high concentration? Inside the cell or inside the water? That means inside the solution, not is the water. Don't get confused. Solution, high concentration solution. In a beaker, high concentrated solution present. In high concentrated solution, placed the normal cell. Normal cell, which means low concentrated cell.
Yes.
Now tell me, water flow inside or outside?
Where the concentration is low, it will move.
Water move from low concentration to high concentration or high concentration to low concentration.
Low to high concentration.
Right, low to high concentration. In this diagram, where is low concentration?
Inside the cell.
Right, inside the cell, low concentration. High concentration inside the beaker, right?
Yes, sir.
Now water flow cell to the out, right?
Yes, sir.
Right. What happen if the water move out? Now this is the full-fledged cell, bulged cell. This is plasma membrane. Actually, when the cell filled with water, with vacuole, filled with water, actually water stored in vacuole. As a result, this plasma membrane firmly attached to the cell wall. They are actually not separated. They are closely associated. But what happened after loss of water? Here the loss, this is by water only. This is actually, for example, this is vacuole. This vacuole filled with water. What happen if you remove the water from the cell or from the vacuole? What happen? The cell bulges out or shrink?
Shrink.
Yes, cell shrink. As a result, cell wall and cell shrinks like this. Cell shrink like this. Now, cut the cell wall. You selected the plasma membrane. This is the mechanical method. Is it clear?
Yes, sir.
Right. But this mechanical method, there are some disadvantages. First of all, this method is highly critical, expensive, skill based. And skill based, high. This is very difficult because, and that is one method. And another one is, already told you, this is not for all cells. Only large and vacuolated storage tissues. What about non-storage tissues? Other cells, the other cells, can we isolated mechanical method?
No, sir. Enzymatic
No.
method.
No, that's why this is not applicable. Because of all cells from the all cells we cannot get protoplasm, number one. And also this method highly skillful and expensive also. And it take more time. That means you placed in hypertonic solution, after shrink, and to cut the with specialized scissors under microscope, with under the microscope. So this is the very risk. And second method is enzymatic method. In name itself, enzymatic. That means simply use the enzymes cellulase and pectinase. I already told you in cell biology what is cell structure, cell wall components. What are how the cell wall made up of? How what are the cell layers, cell wall layers? The first unit itself already told you, explained in cell biology genetics. Cell wall layers. Hello?
Yes, sir. Cell layers.
Cell wall layers and cell wall chemicals.
Yes.
Cell wall layers and cell wall chemicals. Tell me. Cell wall layers and cell wall chemicals. How many layers? Simple. 3 layers.
Yes, sir.
First one, middle layer, either side. Let me change the color. Blue color, primary layer. If when it is matured, and secondary layer also present. This is the cell, right?
Yes, sir.
Second cell, another cell. So, for example, this is the A cell and B cell. For A cell and B cell, what is the connectivity? Where the connectivity? Which layer connect the A and B?
Primary.
Is it connection? Where is the connection here? This is primary and this is primary. But the middle
Mm-hmm.
layer connection, this act as a cement. Act as a cement. This is middle layer, right?
Yes, sir.
So mainly middle layer chemicals, calcium, magnesium, pectate, calcium, magnesium pectate. Calcium, magnesium pectate. And primary layer made up of cellulose, but very rarely form the secondary layer, right? But major middle layer and primary layer. When reach maturity stage, then only the secondary layer also form, right? Sorry, this one, blue color primary. If you want to separate, if you want to separate the cells, if you want to separate the cells, which enzyme utilized here? So, okay, this is primary and green color secondary. What about the next, after secondary layer, next portion is, hello?
Yes, sir. Chemical constituents.
No, what I'm asking here, in cell structure, first middle layer, it is connection between both cells, middle layer. After that, blue
Yes, sir.
color primary layer of the either cells. Next is secondary layer. If it is present, secondary also, but it is not an important one, secondary. But after secondary layer, what is the next portion, next component? What is present after cell wall? This is cell wall. This cell wall. After cell wall, what is the next component? Hello?
Cell organelles.
Next, what cell organelles covered by? Thus, all cell organelles covered by. Just now we discussed what is the next layer of the cell wall. What is this? Are you able to see this diagram?
Yes, sir.
This is cell wall, right? This is the wall. What about this one? This round.
Nucleus, sir.
This is the nucleus. This one nucleus. This one, this layer.
Okay.
What is this? Simple. This layer is plasma membrane. This entire content called as protoplast.
Yes,
So
sir.
what
Yes,
is the
sir.
next to the cell wall? The next to the cell wall is
So
only
plasma membrane.
right plasma membrane and further components. That's totally
Yes, sir.
considered as a protoplast. Now protoplast. Here the protoplast, A protoplast, here the B protoplast. A cell protoplast, B cell protoplast. If you want to separate this protoplast, what will you do? Simple. First, you remove the middle
Middle lamella we will remove.
lamella and also dissolve the primary
Primary wall.
wall. If you dissolve the middle lamella and primary wall and secondary wall, if you dissolve, what are the rest of the portion? Rest of the component? Now cell wall completely dissolved. After cell wall dissolved, what you
Then
can
plasma membrane.
write plasma membrane with protoplasm.
Yes.
The protoplasm separated. This is the easy method because in this method just add some enzymes. The first middle lamella mainly made up of pectates, calcium magnesium pectate. That's why on the pectate the enzyme name pectase enzyme. And primary layer made up of cellulose, hemicellulose. That's why the act by the enzyme named cellulase and hemicellulase. So mainly two enzyme pectinase, cellulase enzymes. If we apply to tissue cellulase, cellulase and pectinase, both enzymes can dissolve the cell wall and get separated the protoplast from that. Now we can select the protoplast, and this is the and and it is less expensive and it takes short time. And one disadvantage also in this because sometimes the A and B both are mixed. We cannot separate. That is the one disadvantage of this enzymatic method. Is it clear?
Yes, sir.
Right. Next one. Let's see the methods of plant tissue culture. Methods of protoplast culture. So it may be where it is marker, just wait. Liquid culture we can use. Agar culture also use. Droplet culture use. Co-culture, hanging droplet culture, immobilized and bead culture, and feed layer technique. These are all utilized. The final one, feed layer technique. These are utilized for the culturing methods. First, let's see the liquid method. The liquid culture, we can use the early development stage of protoplast. The first, after separated protoplast, after inoculated into the agar medium, just 2 to 3 weeks in early stage, transfer into the liquid media. Because it allows the dilution and transfer of protoplast easily get divided in liquid media. So first of all, this growing colors or glowing protoplast required some nutrients. The nutrients easily diffused in from liquid medium, and also it should exchange the gases also. If we use the solid medium in the very beginning stage, how can gases exchange? from the colorless nutrient medium? It is impossible. But if we use the liquid medium, liquid culture method, as it a liquid, in liquid gases easily exchange from the nutrient medium to the developing colors. That's why in very beginning, we utilize the liquid culture media. After colors formation compactly, now the utilize the agar culture medium. Agar culture medium most frequently used sulfide the protoplast culture media. And also this culture media temperature at 45 degrees centigrade and placed in small petri dish. Placed in small petri dish. This is culture media. Next, droplet culture. Droplet culture suspending protoplast in liquid culture media. If we use the liquid culture media placed on petri dish in the form of droplets, that means, for example, this is the liquid medium. When the liquid medium, actually always liquid medium placed in orbital shakers on the liquid medium, here protoplast, blue color protoplast. So this blue color protoplast, when we placed in orbital shakers, this protoplast due to shaking separated into small pieces. Now with help of the droplets, we separated the protoplast with help of droplets and placed in another medium. This is droplet method. So from the liquid medium, protoplast picked up as a droplet and placed on another medium. So this is droplet method. And another one is co-culture method. Co-culture method means protoplast of two different plant species. We can select two different plant species. One is slow growing, another one is fast growing. If we select during the culturing process, one is take more and more time, one is less time. In this situation, we select the both slow growing and fast growing, both mixed. That is called co-culture. So this type of culture is advantageous since the growing species provide the growth factor, because, for example, this is growing culture, blue color and the slow culture, slow development. Why it is slow development? Due to lack of growth hormone, growth promoters, it is slow. Why it is fast? Because deposition of the growth factors. If we mix the both, that means fast and slow. What happened? This both are mixed, meaning the growth regulators also mixed as well. As a result, the slow also become fast. That is the advantage. And hanging droplet method, that is not an important one. And bead culture and also feed layer culture. Feed layer culture, just wait. In feed layer culture, in many cases it is desirable to reduce the planting density. Reduce the plating density. Then feed layer consisting of X irradiation, non-dividing but living protoplast are plating an agar medium, and this layer, the isolated protoplast are placed in thin layer of liquid medium. That means simple, this feeder layer, this is the placed on the agar medium. That means we can place it on agar medium, and also we can place it on liquid medium also as a layer, as a thin layer. Here the living but the non-dividing protoplast provide necessary growth requirement for the isolated less number of protoplast. This method only utilizes for less number of protoplasts. So this is the feeder layer technique, but most probably utilize the liquid medium and agar culture medium. This is the most properly used methods of the protoplast culture. So this is protoplast culture or somatic hybridization, right? Is it clear?
Yes, sir.
Hello?
Yes,
Next
sir.
one. Right. Organ culture. Organ culture. Here organs may be root or shoot or leaf. Anyone act as a organ culture organs. So if you see the organ culture first, let's discuss about root culture. In root culture, we selected, just wait, let me take the marker. Radical tips. Select a radical tip as a explant. Where is radical? Radical present in the embryo. For example, this is the seed, and embryo, embryonic axis, cotyledons. So this is the radical tip. This is shoot tip. We selected explant only radical tip. Aseptically, a germinated seed and liquid medium. In liquid medium can grow independently under the controlled condition. I already told you the controlled condition very beginning. So this root culture first introduced by Cotte, German scientist, and Robbins, USA scientist in 1922. Right? And so root culture can be successfully initiated from the excised radical tips. The root tip cultures are generally maintained in moving liquid medium. Which medium utilized for the root tip culture? Only the liquid medium. In the culture, root tips are induced to grow like that of root system of an intact plant. So finally, clones are formed. A clone of excised roots can also be established from the single root culture by repeatedly cutting and transferring of the main root tips or lateral tips into fresh medium. In every subculture at the interval, different period, last measure the growth of the foot, for growth of the root. So during the culturing process, we can subculturing also this root culture. We can subculture it many times, different type of times, to so that we can get more and more plants. The use of the root culture, root tip culture increase our knowledge of carbohydrates, metabolism, role of minerals. Sorry, not cutting minerals and ions, vitamins in growth of the roots. So how the minerals utilized, how the ions utilized, how the vitamins utilized for the growth of the roots, we can observe. And clones are ideally suited for the study of the effect various compounds of root growth. And we know the leguminous plants for nitrogen fixation. Root culturing of leguminous plants provide ideal system to study the procedure nodule formation. Not only nodule formation, and also we can understand the how to fixation nitrogen, how to fixation atmospheric nitrogen inside the plants, inside the rhizome, inside the leguminous plants. This is done by Raggio and his colleagues. So these are the importance of the root culture. Like that, another one is shoot culture. Shoot tip culture. Same like this. Here the root tip explant, but here just before root tip. Root tip is the explant, but now the shoot tip culture. That means shoot tip is the explant. So we selected the shoot tip and placed the agar medium, and later it provide the colors and embryonic cells. After that, we get a new plant. So this is the most important thing is actually the one important thing is we can get virus free plant through shoot tip culture, because the virus can grow everywhere. Virus can grow everywhere except the root tip. Sorry, except the shoot tip. Except the shoot tip. So if we selected the shoot tip as a culture, shoot tip as a culture, already the shoot tip have the property of the not to permit the any virus. So from the shoot tip we can get the normal and the whole plant. We can develop the whole plant. The same property carry forward to this plant also from each and every part, because it is complete development shoot tip. The shoot tip properties not to permit the virus, any virus. From the shoot tip, the whole plant developed. So now the characters carry forward to the whole plant also. Now this plant, virus-free plant, get developed as a virus-free plant. That is the most important one is of stem culture or shoot culture, not a stem, shoot culture. And next, right, leaf culture. The same leaf in the same like a root and shoot. In leaf culture also, the young leaf or leaf primordia selected as a explant and transfer into the medium, transfer into the nutrient medium. After that, we can get a replant. The same process. The uses of the leaf culture is a valuable technique to study effect various nutrient, growth factor, environment condition of leaf development under controlled condition. So that is the most important one is, and also we can study about chlorophylls, protoplast, and some of the synthesis of the leaves. Also we can study with help of the leaf culture. And also we can observe the fern and leaf primordial culture to study sporangia. How the sporangia formed, for example, sporangia develop. Sporangia develop means, for example, in ferns or gymnosperms. Ferns, this is the leaf. This is the leaf. The leaf with sporangium, either micro or macro. To culturing this leaf, we can study about the sporangium development. So these are all the important of leaf culture. And the next class we can discuss. We will discuss reproductive organ culture.
How
Reproductive
many
organ
classes
culture.
we are having, sir?
I think around three. Three completed and three are remain.
Okay. It is these classes have started from, sir, because we didn't get the notification, sir. Last two classes.
Actually, they sent to in group also or in website. Are you not follow a website?
No, sir, I didn't saw.
Do you have
Uh,
a WhatsApp group?
yes, sir.
Yeah, I think I am in the group also. I will
Yes,
share
sir.
the link.
Okay, sir. Okay.
Dates also. Okay.
Okay, sir. Okay.
You
Thank you,
said
sir.
your friend also ask them to join in further classes.
Okay, sir.
Okay. Thank you. Thank you all.
Thank you.